aware, the Berkeley Brain Initiative, through a panel discussion that I attended, and there were 3 very noted researchers there, and when I asked one of them if they could speak, Dr. Ball was gracious enough to step up. So we're fortunate in the Bay Area to have such great centers of research going on, looking at neuroscience and the brain and from all different angles. We're going to get a perspective of that today, of some of the work that's going on at Cal. By no means is it exhaustive, but it should give you a glimpse into some of the fascinating fundamental research that's going on. So Dr. Ball, please take it away.
Great. So, hi everyone, my name is Robin. I'm a lecturer at UC Berkeley. I also work in the lab of Feilud Isakov, and we're in the molecular cell biology department and also the Helen Wills Neuroscience Institute. So today I'm going to tell you about some of the really cool research that we're doing at UC Berkeley to try and advance new technologies to study the brain and to study neurons, the cells inside the brain. And so this image that you're seeing here is a movie of a fruit fly ventral nerve cord, which is kind of like the spinal cord. And the things that you see flashing there are actual nerve cells that are activating as we speak, as this larva is trying to move across the microscope stage. So I'll come back to this towards the end of the talk today. This is some of the things that we're going to be talking about. So first I just want to introduce you to the brain, to the nervous system. So as you know, the brain is very complex. It's this big mushy ball here, very bloody looking, lots of blood vessels surrounding it. But if we zoom in inside of that, we see that there's lots and lots of individual cells. So there's like 8 billion neurons, those are the nerve cells, and 100 trillion synapses, which are the connections between nerve cells. So in this image here, we're seeing a brain, and they've imaged some of the long processes that come off of the cells when they're trying to communicate to other cells. These are called axons. And so as you can see, there's a ton of connections between like the left and the right side of the brain, and the top and the bottom. And this is just to illustrate again that this is very complex. How do we make sense of all of these cells? And this is another image here of just a little piece of a mouse brain. And the spots that you're seeing in green, those are the cell body of these neurons. And then again, they have these processes that are going off everywhere to try and communicate with other parts of the brain and other parts of the body. So it's very complex. That's kind of the summary of this slide. So how do we make sense of this? We need to get new technologies in order to try to understand how the cells are communicating and how they're functioning. So before I get into the technologies, I wanted just to do a little bit of terminology here, so I'm going to be using some words, and I want to make sure you understand them. So this is a nerve cell here. It's called a neuron, and it has a cell body. That's where the main parts of the cell are located, like the DNA, things like that. And then it also has all of these extensions coming off of it. So the ones on this side are known as the dendrites, and they receive signals. So other nerve cells will release a chemical onto them called a neurotransmitter, and that tells that neuron what to do. And then if the neuron gets excited and gets activated, then it has an electrical signal that travels down this longer extension here called an axon. And that can be very long. It can go all the way from your toe up to your brain. So we have some nerve cells that are actually that long. And then it will release a chemical messenger to the next cell along. And then that again is called a neurotransmitter. So this could be onto another neuron, or it could be onto like a muscle cell to tell it to contract, or your heart cell, or whatever. Okay, so that's how neurons function. Right, the brain is really complex. How are we going to understand it? So we're developing these new technologies, and this is part of what's known as the Brain Initiative. This was started in 2013 by President Obama, and it's a way to fund new technologies to advance our understanding of the brain. So some of the key goals of the Brain Initiative are shown here. So one is to advance technologies to study neuroscience. I'll be talking a lot about that. Also to facilitate dynamic brain imaging to see like the actual brain in action in a human. To explore the functionality of the brain and to connect that to behavior. That's one of our main goals. And then also to advance consumer applications, which I'll talk about as well. So lots of the grants that have gone out for the Brain Initiative have gone to UC Berkeley researchers. And I'm just going to talk about 3 of those. So we call this the Berkeley Brain Initiative. So these are the 3 that I'm going to go over in more detail. The first one is David Feinberg, and he is trying to advance new MRI technology, so to study the brain as a whole, and he wants to increase spatial resolution by 30 times. So I'll talk about that one first. The second one, we're kind of going to zoom in from the brain to the individual neurons. And this is work by Jose Carmena, and he's trying to make new microscopic sensors that you can put into the brain and leave them there for like a decade and record electrical activity from neurons in action. So we'll talk about that. He calls it neural dust. And then the third one is work from our lab, so the Isaacoff lab, along with some other collaborators, and we're trying to look at the activity of neurons in animals as they're behaving. We're trying to figure out, make correlations between activity and behavior. Okay, so I'll talk about those three in order. And for all of these, I'm going to introduce first kind of the old method of doing things, and then we'll see how the new method is better. So how do we study function in the brain? So in the 1800s, lots of people were interested in how the brain worked. And this was usually work done by neurologists. They had these patients who had strokes, and they had some sort of deficit afterwards. So a famous case are these patients of Dr. Broca. So they had this thing called aphasia. And aphasia means that you have a hard time with language, either processing language or speaking language. And this happened after they had strokes, and so there's damage to the brain. So he studied them, and then he waited until they died and did an autopsy of the brain. And as you can see in one of these brains here, it's pretty clear that there was damage to this part of the brain. So he concluded, okay, the front left area of the brain is important for language. So that's a good start, but this is a huge area, and this is in a patient who had deficits. We like to study normal individuals who are actually doing language and see like which parts of the brain are active. So the new method, and this is pretty common these days, is functional MRI or fMRI. So it's using an MRI machine with magnetic fields, and instead of just getting a static image of the brain, you're getting an image of the brain as it's active, and like which parts of the area are getting more blood and more oxygen flow, and then those are the areas of the brain that are active, while the subject is doing some tasks. So they're like lying in the magnetic field, and they're like looking at visual cues or auditory cues, and then you can see which parts of the brain are active, or light up, as they like to say. So this has been done to study language as well. This is not something I'm going to talk about much, but I just wanted to point it out because it is a new study that came out of UC Berkeley. This is from the Jack Gallant lab. And they had normal people in an fMRI, and they were listening to podcasts, just kind of passively listening to the podcast. And they imaged the brain while they did this. And they were able to correlate which parts of the brain were active when they heard certain words and certain phrases. And so they could make what we call a semantic map, or a map of the brain for different word meanings. So which parts of the brain are processing those words as you hear them. And so from this, we can see that actually the whole brain is active during language processing. So Broca was a good start, but we definitely need more studies of dynamic brains to figure out what's going on. Okay, so that's fMRI, and it's pretty good, but we want to make it better. Okay, so again, this is work by David Feinberg in the psychology department. And so fMRI has a pretty good spatial resolution. What that means is that it can pick up individual like 1 millimeter pixels or 1 millimeter boxes within the brain. So that's pretty good, but it's not great because in that box there's like 50,000 of these neurons, and they're probably not all active at the same time. So we'd like to be able to see the differences that are happening on a smaller scale. And so he has this new technique. He's calling it MR cartography. And it's basically just going to increase the resolution of fMRI about like 30 times. And the way it works, I'll just briefly explain it, is that he's going to use really, really small, sensitive coils, which are the sensors in the machine that are picking up the changes in the magnetic fields. And the reason why we haven't done this before is because if you use small coils, you only image the outside of the brain. And so most people, when they're doing MRI, they want to see the whole thing. And that's what's shown in this image here. So this would be like if you had a large sensor, you're picking up all the information from like the back half of the brain. You get pretty deep in there, which is great. But the thing is that you have bad resolution. So if you use smaller coils and you just pick up the information from the back part of the brain, just kind of on the outside, you're going to get finer resolution. And for these kind of studies, it's okay because it turns out the outside of the brain, which we call the cortex, is the area of the brain where we do language processing and visual processing, auditory, like all the kind of cognitive things that you really want to study. All of that's happening on the outside of the brain. So you don't really need to worry much about what's going on inside, at least for these kind of studies. So that's the idea there. So this grant is going to help to build these on the UC Berkeley campus. And then we can also use these for medical studies as well, not just for trying to figure out how the brain functions. So for example, you can use fMRI to look at patients who have epilepsy as they're having a seizure. And so seizure areas are areas of the brain that are very active, they're too active. So you can see that with fMRI. So this is an example using the old method of fMRI, and you can see like these areas that they've colored here are the areas that were very active while this person had a seizure. So you can say, okay, the front part of their brain is active, and if they need to have surgery, then maybe that's an area that you could take out or alter. But still, the resolution isn't great. So it'd be really good if we could do this using the new MR corticography to get really fine resolution of where the seizure is occurring. And you'd be able to help these patients, maybe. Another possible use of this is to figure out which parts of the brain have been damaged by a stroke or by a traumatic brain injury, like a concussion. So again, you could look at which parts are less functional than they were before. Okay, so that's MR corticography. And I should say, I guess if you have any questions, you can ask me afterwards or during the potluck. Yeah, okay. So the second method that we're going to talk about here is this thing called neural dust that Jose Carmena is working on. So in the last subject, the last area that we were talking about, we were doing imaging, right? We were imaging the brain. You didn't have to like have anything open, just imaging the brain. But now, what if you want to actually record directly the electrical activity inside of those neurons, so that electrical signal that's traveling down the axon? Well, we can actually do this now, and they do this in patients who are having brain surgery for some reason. Like maybe they have a tumor that needs to be removed, or they need to have some seizure area of the brain removed. And so you can use this crazy contraption here. It's actually just a whole bunch of electrodes that are sticking into the brain. And the patient is awake during these surgeries because there's no pain receptors in the brain. So they're awake and fully functional, and you can talk to them, and as you talk to them, you can see which parts of the brain are active and actually record from the neurons. That's really cool, but there's only a few patients who you're doing this to, and you can only do it for a limited time, right? Because they have to have their surgery and then recover. And this is kind of the new method of doing it. Instead of sticking electrodes into the actual brain, you can have them kind of lying over the surface. So lots of researchers do still do these kind of studies, and we get pretty interesting information from it. But wouldn't it be great if we could record from them for an extended period of time, like close up the brain, and keep on recording for years later? And so that is where neural dust comes in. So this is a tiny, tiny sensor, and this is actually bigger than what they want to get to. Eventually, they want to get it to half the width of a human hair. So very, very small sensors, nanosensors, that they're going to implant into the brain and just leave there. So you close the patient back up, and you can leave it there, and they're still recording the electrical activity from just a couple of neurons as the subject is moving around. So that's the idea with this new method. We don't really need to worry too much about how it works, but I'll just kind of orient you to this diagram here. So this is the brain. This is the cortex. Remember, that's kind of the important part of the brain, the one that we're interested in seeing how it functions. And the little dots here would be the neural dust. So very, very small, little tiny sensors. You just kind of drop them in on top of the brain, I guess. And then there'd be a little transceiver on top of the membrane that surrounds the brain. And so you're receiving the signals from that sensor, and then that sends signals to a bigger transceiver that's outside of the head that can just sit there. But it's pretty, once you've put them in there and closed them back up, it's pretty non-invasive. And this is showing a prototype, so they've put it, this is the sensor here, on top of a nerve in a rat, and it functions. It can record the activity of that rat as it's moving around. But again, the future goal is to make it much, much smaller, so 50 micrometers, that's again half the width of a human hair. It will be able to record from just a few neurons, so rather than 50,000 in the fMRI, we're going to just be recording from a few here. And ideally, they want to be able to leave it in someone for up to a decade. And you wouldn't have to worry about infections or anything, because you've already closed up the surgery. And then the reason why we're doing this, in case you're wondering, we're not just doing experiments on people just for the fun of it. We want to help people who are paralyzed using these brain-machine interfaces. So I'm going to talk about what that is. This is a really cool subject. And this is a collaboration between Jose Carmena and Michael Maherbiz in the electrical engineering department. And they also just got a really big grant yesterday or Friday. So this is a really big topic these days. So here's how it works. So someone is paralyzed because they've had a spinal cord injury, right? So the spinal cord's been broken. That means that the signals from the brain can no longer go down to the muscles and control muscles, right? So they're paralyzed below where that injury site occurred. But their brain still functions totally fine, right? Nothing's wrong with the brain. And so the brain can still send signals like, I want to move my arm or I want to move my leg. Those signals still work in the brain. And so what you could do is put the neural dust into the motor area of the brain. You know where that is, it's like right up here. And then the neural dust would be recording what's happening in the motor cortex, the motor area of the brain. And then it sends that signal to a computer, which tries to interpret that pattern of activity. And so the computer learns, like, when this particular type of activity happens in the motor cortex, that means this person wants to move their leg. Okay, so then the computer knows that, and maybe the computer is attached to a prosthetic leg or a prosthetic arm, and it will move that for you, as if it was your own leg. Or, even better, you could put electrodes into the muscles, and the electrodes will stimulate the muscles and cause the muscles to contract. Just as if you had sent the signal from your brain and your spinal cord to the muscles, the computer is going to kind of act as your spinal cord. It will interpret what's happening in the brain, send a signal to the electrodes that are in your muscles, and your muscles will contract. And so this has actually been done not using neural dust, but using electrodes that sit in the brain. So there's like wires coming out of the scalp. And that's pretty invasive, could lead to infections. And so neural dust is really going to help improve brain-machine interfaces in the future. So that's fine. It's a really interesting subject. Okay, so we've gone from like kind of whole brains to looking at a few neurons inside the brain. And we're going to move on to individual cells, individual neurons in animals. Okay, so how do you record from an individual neuron? So the old way of doing it, which we still do these days, is to use an electrode, which is this thing here. So this is not normally found in the brain, right? So you stick it into a cell, and you record that electrical activity inside of that cell. Now, in order to do this, you definitely have to dissect open an animal to get access to the brain, to the neurons. Maybe you take the cells out and are just doing this in a dish. So we've learned a lot about how neurons function by doing this, but it's not very realistic, right? We want an animal that can actually walk around and see how the neuron activity correlates with behavior. That's the idea here. So the new method is that you can use fluorescent proteins. These are proteins that glow a certain color, and they get brighter when the neuron is active. So we can put these into our animals, into particular neurons, and see when those neurons are active, because it will get brighter. We can just image it. So this is the image that we saw on the very first slide. So again, this structure here is called the ventral nerve cord, and it's like the spinal cord of a fruit fly. And actually this is a larva, which is, you guys call it a maggot, but we call it a larva. Okay? So you can see them by eye, but they are very, very small. And you can see through their skin, their cuticle, it's transparent. So this is a totally intact larva here, and we can see that there's waves of activity that are spreading through this ventral nerve cord, right? So that those white areas that you're seeing are active neurons. And this is while the larva is trying to move. Okay, so I'll show you this again, and we'll talk about how this actually functions in more detail here. So I have to go over a little bit of anatomy, just so you understand what's going on. And this is work done in the Eisenhower lab, again, and then the person who's kind of in charge of it is Zach Newman. He's a postdoc in the lab. So this is what the larva looks like here. So it is transparent, and the kind of the white stuff that you're seeing here, those are the guts. So they have lots and lots of intestines. All they do is eat, basically their main function. So let's look at a little cartoon of this. So in this cartoon, I've just removed out the guts. We don't need to see that. And we're just going to open it up so that you get a better view of what we're looking at here. Okay, so this thing in the middle is the brain. That's the ventral nerve cord. And then these things in green are the body wall muscles. So they're helping the larva move. So it kind of crawls along like an inchworm through the food as it's eating. And so from the ventral nerve cord there, you see like all these black lines that are coming off. Those are axons. Remember, those are the long processes of neurons. They're sending electrical signals to the muscle, and they're releasing neurotransmitter onto the muscle, and then the muscle contracts. Right? So it's controlling muscle contraction. And then let's zoom in on one little area here. So we can see that each one of these is a muscle here, each one of those green things that you're seeing, and it's stereotyped, so all larvae have the exact same muscles. So they're all numbered, we can find them again. And then the black line here is the axon coming from the ventral nerve cord. And again, when it is active, it will release neurotransmitter onto the muscle, and the muscle will contract. And that happens at a synapse, which is a connection between two cells. So here it's a connection between a neuron and a muscle, and we call that synapse the neuromuscular junction. So the neurons releasing neurotransmitter, and then muscle as we're releasing it onto a muscle. So that's basically what this structure is here. You don't really need to worry about the 1S or 1B. There's just different types of synapses that are made. And so again, they're going to release neurotransmitter onto the muscle, and then the muscle will contract. So how do we record activity of this synapse? Like, how do we know when it's active? Well, we have this flesh-up protein that is not normally found in fruit flies, and so we can genetically alter them and put the protein into them. And we made it so that it's localized in the muscle right at the synapse. So all this part in green is the green flesh-up protein. So all this part in green is the flesh-up protein. And then, as I mentioned earlier, it gets brighter when it's active. So when neurotransmitter is released, then that muscle will get active, and we'll see this green get brighter. And all of our images actually are in black and white, so when you see white getting brighter, that means that it's a synapse that's active. Okay, so that's the idea here. And it works in larvae because they're see-through again, as you can see right through them. So this is a larva just moving across the stage. You can see it kind of moves like an inchworm. Actually, the white spots here are not really the active synapses. I'll show you that in a second. It's microscopic, so we have to zoom in on that to really see it. But just so you can get an idea of what we're looking at. And you can see that if we try to image that, we have to have a microscope that moves along with the larva. So instead, what we do is we stick the larva into a chamber where it can't escape. So this is the little chamber that we made. It's just stuck inside of there. But it is still moving, right? So it's alive. Its muscles are contracting, but it's not going to escape off the edge. So it's much easier for imaging. So we're just going to zoom in on one area here to look at the synapses that are being activated. Okay, so I'll just let you look at it for a second, and then I'll explain it. Okay, so you can see like white things showing up there. All right, so there's kind of two different things that you're looking at here. One is that you can see kind of whole muscles getting brighter, and that's just because the muscles are contracting, and so they're getting smaller, kind of more condensed, and so it shows up as a wider area. And so you can see like all of these muscles, all the muscles like on the left side contract first, and then it gets a wave that travels through that. We're also seeing those really bright white spots, and those you may recognize as the neuromuscular junction. So those are the synapses. So they're dark when there's nothing happening because they're at best, and then they get brighter when that flesh protein gets activated. Right? So those are active synapses, and we're just seeing it right here live in this larva as it's trying to move. Right? So the synapses get activated first, and then the muscles contract. So it's pretty cool that we can do this in the larvae as they're moving around. But what is the whole point of this? Well, we're just kind of interested in how synapses work in general, but it is definitely applicable to mammals and to humans. So fruit flies have the exact same kind of synaptic machinery as humans do. So our synapses function just the same as fruit flies do. We have the same proteins, the same kind of genes, and so on. This image that you've seen in green is some of the neurons that are in the brain of the fly. This is a lot simpler to study than studying it in a human, obviously, and even in a mouse. So that's why we like fruit flies. And then it is useful for studying diseases as well, because the genes that cause diseases in humans are found in fruit flies too. Right? They're not going to necessarily cause the same type of behavioral problems in the flies as they do in humans, but it's going to cause the same sort of problems to their synapses. So what we can do is we can very easily impair individual genes in the fly and then go back to our imaging and see how the synaptic function changes. So how does that activity change and so on. And that will tell us more about the function of these disease proteins. And so it's really useful to study the fly neuroscience and then apply that to mammals and then eventually to humans as well. And actually, just by chance, one of the students who is working on this project is here, Julia. So if you want to learn more about it, you guys can talk to her maybe a lunch. And if you're interested in knowing more about the kind of research that's happening at UC Berkeley, I put up the website address here, and they list seminars, so anyone is free to go to any seminar you want. It's open to the public. And those are usually speakers coming from other universities to come talk. And then there's also a news area of this website too that tells you about all the really cool research that's happening, new papers that come out, and so on. Okay, so that's it. Thanks for listening.